presentation. And with that, I'd like to open this up to JP to begin his presentation on getting connected to your data, a reproducible workflow for data wrangling. Thank you, JP.
Thanks, Wanda. I appreciate that. Very lovely welcoming. And hello, everyone out there in cyberspace. I'm Jean-Paul. Just so you have a means of being able to reach out to me, I have added my email address here for you, and also my Twitter handle, because you may want to go check out some of the stuff that I'm interested in on Twitter. A lot of it's retweets of scientific related things, data, tutorials. There's a wealth of information out there for that kind of stuff. And also, so you know, there is a recording and slides for this talk, which can be found right here at the link below. Oop. And here we go. All right. So it's exciting to imagine bringing your data product to life. And bringing data products to life with researchers is one of the most rewarding parts of my job. So what I hope for you to leave with today, having learned, is a few things. I want you to know what data wrangling is. I'll talk about a dual meaning of data wrangling in hopes of clarifying what I mean when I'm talking about data wrangling. Also, I'd like to introduce you to the idea of tidy data, which has a general meaning describing a structure of data to consider when transforming your data for your intended goal, but also a very specific context related to software that can be leveraged for tidying data and using tidy data. And once we've made it through all that, we'll talk about some data workflows, particularly one of my data workflows, and we'll look at some considerations on how to make your overall data wrangling effort less time consuming. So every time I look at this dashboard, I pause. This is a deeply impacting work of data. It's arguably one of the most famous dashboards of modern times. This dashboard framed a view of the growing concern that has become the pandemic we are in. It's the inspiration for many other similar dashboards. And however, despite all that, behind the scenes of this dashboard is multiple data sources being combined into one format that is the source data for what we see. And if you're interested in looking at all the sources, the links below take you to all the sources of data and the data itself. Here's another dashboard. This time it's actually one that I worked on, not just looked at. I spent a lot of time looking at this one. And this project started with a researcher who was interested in finding out more about how to create a Tableau dashboard. And I have nothing against Tableau dashboards. They're great, but we just ended up going with an alternate option, that being our Shiny dashboard. Shiny is an R package. So the data for this dashboard came from Epic, REHR, and it was accessed through a request from Research Harbor. The goal of the project was to create a dynamic visualization that can inform nurses about key information relevant to their procedures and whether that would improve outcomes if implemented in the clinic. As with the COVID dashboard, the researcher was faced with having to figure out how to transform their research data from the output, from where it came, to the input required by the software running the dashboard. This brings me to the point of value. Since you're here at this talk, I know it's because you're interested in extracting value from your data. We can categorize this value into two types along a temporal dimension: near-term value and long-term value. In the near term, you likely have a sizable list of questions that you want to answer using your data. Some of these questions might be vague. For example, did a nurse document assessing the site of a central line insertion? Other questions might be more specific, like how many central lines were inserted over a specific span of time. Beyond the list of known questions related to near-term value of your data is the optimism that your data has a greater potential long-term value. Can you use it to decrease the number of hospital-acquired infections, for instance? So another dimension of value to consider is how that value will be delivered into your organization or audience. Will value be delivered directly through systems that can take automated action based on data as it's processed? Or will value be delivered indirectly by empowering people in your organization to take a different course of action than they otherwise would have? When data provides value to your organization or audience by influencing people's decisions or inspiring changes in processes, for example, attending a talk about data wrangling, we consider this indirect value. Data provides value to your organization by feeding automated systems. For example, Netflix's recommendation system. That's considered direct value. So, what's stopping you from answering the questions? The most common answer mentioned is time. You know the questions, you know how to answer them, but you just don't have enough hours in the day to get your data into the right form. So that brings about data wrangling. Learning to do this well is the best investment you can make in yourself as a data scientist or someone working with data, because it allows you to realize the value in your data. So you've probably heard some of these terms like munging or transformation or manipulation. All of these terms are terms used to describe data wrangling, a process that, when it's distilled down to its simplest understanding, it's two things: making your data suitable to use with a particular piece of software and revealing information. So what's the process for wrangling data, or a wrangling workflow? Some people think that wrangling data refers to the step right here in the middle, the transform profile step. Some people will say that wrangling refers to all the steps. And to be honest, they're both right. Fundamentally, remember, data wrangling is a process involved in transforming or preparing data for analysis. Breaking down this workflow step by step, we see step 1, or step 0, is accessing data. This is all about where your data comes from and how it's organized. So it could be simply someone providing you a raw data on a USB drive, downloaded or scraped data from a website, or even something as complex as programmatically or interactively accessing data through an API query. So you're really thinking about also the structural organization of the data and how it's organized on your file system. So remember that dual meaning of data wrangling I talked about? This step is where that duality comes into play. Very often, guidance and instruction and reference to data wrangling refers to the iterative process of transforming and profiling. Transforming, where we're changing the form of the data, adding new values, fixing irregularities, and profiling, where we're summarizing the values of variables across records or validating individual records. Now keep in mind that this is the step that most people think about as data wrangling. It's a intensive, involved process of cleaning and rearranging, and due to time constraints, we're not actually going to get into some functions or processes where we take some data and transform it ourselves. But just know there's no shortage of guides for this process for whatever way that you do it. And I'm going to show you in a few slides some examples to inform you of some transformations to make. Now, at the end of your workflow, you get to the publish stage. This is where you have your data structured for the software that you intend to use. This could be a finished data set that's used for the data product, and you just plug it right in. Or it could be the script that you use to wrangle the data. This could also be a data dictionary or some other type of metadata presentation that's showing you what's going on, the finer meaning of your data set. Now, I brought up that in the transformation step, you're going to be using tools. And actually, you're going to use tools all along the pipeline of wrangling your data. Here's a selection of some tools to consider. We have Python and R. And the way these tools are structured is declarative, and they imply the steps you intend to take in transforming your data, in a way suggesting these are the things you want to do. One big advantage of these particular tools is that at the end of your process, considering you're not doing it interactively, is a reusable script that states all the functions you took in a stepwise fashion to create your output. A disadvantage of these tools, though, is the learning curve for learning how to code. But the advantages for using these tools particularly far outweigh the learning curve, which actually eventually flattens out. So there is a ceiling where you become the consummate programmer. Excel, which everyone probably is familiar with at this point, offers you advantages you all probably already know. The advantages are you probably already know how to use Excel, and it's widely available, so it's more than likely already on your computer. You can see your data set right in front of you, and that's great. However, there's some disadvantages as well for using Excel. There's limits to reproducibility without significant additional effort writing all the steps that you took. And also another issue is that you're directly interacting with the data that you'll be using for your final purpose. And that can leave you open to introducing error. Complementary fact to this is that there's also similar tools like LibreOffice, which allow you to do what you would do in Excel or Word, but without having to purchase software. So you can do the same things as you do in Excel via an open source version. Now, in the realm of data wrangling specific software is a tool like Trifacta. It's a data wrangling tool specifically developed for data wrangling. It's able to analyze your input data and make suggestions to you that you can go step by step in cleaning up your data, and that's very useful. Some of the disadvantages, however, is that it's proprietary and it costs money. Although there is a free tier, it just has a lot of limits on it. And finally is OpenRefine. OpenRefine is a unique tool that combines the power of databases and scripting languages into an interactive and user-friendly visual interface. It keeps track of all your actions and allows them to be applied to different data sets. So it has a reproducibility feature built in. OpenRefine can import a variety of file types, and it's particularly useful for parsing results from APIs. Some of the limits and disadvantages mainly would be size limitations. So you'd have to really get into the system and reconfigure it to allow it to work with larger data sets that are over 100,000 rows. So as a data scientist, hacking your way to an answer is part of the gig. And finding resources to figure out how to do a task when working with your data and unraveling how to use tools is critical. So a guidance that I have for you when looking for a guide or tutorial or watching a YouTube video is to determine if you're seeking general or specific guidance when staging that Google search. For instance, generally you could write in data wrangling with Excel or data wrangling with R versus more specific, distribute the contents of a cell into adjacent columns in Excel, or gather columns into rows using the tidyr package. So the two examples I have here, and there is a third example, but that's a link to a guide that provides you some more resources beyond these two for you to consider. The two examples provide both specific and general advice. So if you were to click on that link, the Microsoft example provides a resource with specific guidance on cleaning your data. Here in this section, the basics of cleaning your data, general guidance. And then below, specific guidance like on spell checking and how to do that, removing duplicate rows, so on and so forth. This, by the way, is a useful checklist to have for you to refer to and considering the different steps that you're going to take in cleaning your data in that transformation step. This cleaning is very much geared towards the transformation step of the data wrangling process. On the other hand, we have this super cool data wrangling with dplyr and tidyr cheat sheet. This offers a guidance on the different steps that you're going to want to take in your data wrangling and transformation process, and also gives you specific examples of the code that you would run whenever you are cleaning up your data. And of course, remember that I'm always available too to bounce ideas off and questions off of. So I'm here for you. Go back to my presentation. Here we are again. All right. Tidy data. Some of you may have heard this term before. When you hear this term, know that what we're talking about is data structure, having your variables, observations or records, and values organized in a particular format. So take a look at these data sets. Data sets come in many formats. We can go through these data sets and examine what they contain. In particular, we can look at the variables in each data set, seen here as tables. First, we have the storms data set on the left. It has 4 columns, with the first row labeling names for each column. In this case, the names happen to be variables of data collected on hurricanes. We have the storm name, wind speed measured in miles per hour, air pressure, and the final column, we have a date. On the right is a data set for cases of TB in countries in the years that you see here, 2011 through 2013. We have one variable, country, which is in the first column, in the first row, but for second through fourth columns, you see a year, and then you have another variable that is embedded into this table, which is a count of cases of TB, we'd say. So looking at these two lines and shapes in these two tables, we see a distinct difference between the structure of the data sets. Storm structure has variables in all columns, while cases has them arranged in various formats. So following these three rules is what makes a data set tidy. Variables are in columns, observations are in rows, and values are in cells. These three rules are interrelated because it's impossible to only satisfy two of the three. So you might be asking yourself at this point, why ensure that your data is tidy? Well, there are two main advantages to tidying your data, or the tidy data structure. There's a general advantage to picking one consistent way of storing your data, for instance, because if you have a consistent data structure, it's easier to learn the tools that work with it because they have an underlying uniformity intended to work with that structure. There's a specific advantage to placing variables in columns because it allows Python or R's vectorized nature to shine. And that just means the way that Python and R deal with the data when they're running functions. So when you're working with visualization software and machine learning algorithms in R or Python, like those I mentioned at the beginning of the talk, to produce data products, those rely on tidy data sets. So keep in mind, this is a non-arbitrary standard for structuring data, but it's not completely unambiguous. So I don't want to leave you hanging, wondering what that case's data set would look like when it's tidy. So here it is. You see now, we have a table with columns containing three variables, and the country, the year, and the number of cases. So one way to make that happen could be a bunch of copying and pasting, and that might work, but that won't work for me, because if I have to do it more than once, I'm a lazy guy. I'd rather not do that. So there's this rule in programming that if you have a task that you do more than once, write a script for it. In R, you could do this organization with one line of code, just like this. This code says, collapse multiple columns into two columns. One, a key column that contains the former column names, and two, a value column that contains the former column cells. And then give me that count, which is the values of the cells. So I was talking a little bit about R, and it's that time now for your R indoctrination. I want to make a note that you may think of tidy data as synonymous with R or part of R, but as I pointed out, it's actually the preferred structure for working with R because most built-in R functions rely on a vector of values, and tidy data ensures that your variables are organized in vectors where all the observations are individual elements of the vector, but we don't need to get into all that technicality. But it is worth knowing that RStudio provides a set of packages known as the tidyverse that are designed to work with tidy data. And there's a subset specifically for performing data wrangling tasks. Those are the ones in the middle, dplyr, and tidyr. And this is going to allow you to work on that transform step. And trust me when I tell you, there is a ton of really in-depth guided tutorials that instruct you how to do that process. Another way that you can actually get tidy data is by using REDCap. Now, REDCap allows you to create data collection instruments and data dictionaries. And those two things work interchangeably, meaning you can use your data dictionary to design your data collection instrument, and your data collection instrument will populate your data dictionary. And the reason I'm bringing up REDCap right now in tidy data is it just so happens that the structure of REDCap is designed for you to inevitably put all of your variables as individual components in your data collection instrument, so that whenever you export your data, it's actually going to be in a tidy format. And that's great. So you can save yourself the step of having to tidy if you start collecting your data in a way that ultimately allows you to export it to tidy. Another nice feature of REDCap is that you can actually use it to de-identify your data as well. Another task that will come up in the data wrangling workflow. I know what you're thinking. Well, what if I don't need tidy data? Tidy data is definitely a great standard to follow, and now y'all know that. And there are cases when making your data tidy doesn't make sense for your project. There are lots of useful and well-founded data structures that are not tidy, and there are two main reasons to use other data structures. Alternative representations of your data may have substantial performance or space advantages, and also specialized fields have evolved their own conventions for storing data that may be quite different to the conventions of tidy data. And those are very strong, relevant, valid points. So if your data does not fit into a rectangular structure composed of observations and variables, if your data does, I think tidy data should be your default choice. But as you now know, there are good reasons to use other structures. Tidy data is not the only way. So how are we going to start saving time with our data wrangling? Let me flash this back up here again so we can reconnect with this idea that most folks think of data wrangling as what's going on in the transformation and profiling stage, where we're cleaning up and changing values, and that very much is the case for data wrangling. But for the purpose of this talk, we're thinking of this model as the process of data wrangling, and just know that each step has ways of informing the preceding step and the preceding step. And that's why we see these arrows. I didn't mention the arrows previously on purpose because I wanted to bring them up here. So data wrangling is iterative. That means redundancy. I alluded earlier to being a little bit lazy, and doing things less repetitively is a huge time saver. So let's think about that. I want to pose some questions for you to think. For reuse, will I repeat this analysis that I'm working on for my data project? Will I want to see anything else from my data? Will I want to add more data to my analysis? Or is there a chance I will retool the data for another piece of software? If you can answer yes to these questions, and others like these, the chances are you stand to benefit from thinking about a programmatic or scripted way of managing your data wrangling. Let's look at some examples of what I mean. So I work with RNA sequencing data, and when working with raw data from the lab that has performed the sequencing, I routinely have to go to their website to download the data. The files I'm accessing are very large, many gigabytes in size, so I can't just scroll through them to look for errors. So it's a standard practice to verify that there were no errors introduced to the file during retrieval. One thing available to me from the sequencing facility when I download the data is a file with a sample name and its corresponding file name, and it contains the data and a checksum for the file. The checksum is an elaborate, unique string of letters, as you see over here under MD5 sums, text file or calculated. And my first wrangling task typically is to verify all my files checksums so I can make sure there was no errors in the downloading process, because during the access, that's one of the things I have to do. So I can go about doing this at the command line, running the simple function mdsums on this file name, and I can wait, the computer calculates it, I hang out, I might listen to a song, I get my results. It's a little bit boring maybe, but after the first couple files, I'm really starting to wear down. Not to mention, I have to create a document where I report the file name from the sequencing facility and its checksum side by side with the file name from my system with the checksum that I calculated. Now imagine doing this like weekly or even daily. This can be a major time drain, especially doing it interactively file by file. So one way to speed this process up and pay it forward is to leverage the power of a script. Now, keep in mind, I know that there's an upfront cost for writing the script, but that cost is minuscule in comparison to the time that you're going to save from spending this process being done by hand. Here's another scenario for a continuation in my RNA-seq project. Right here, you see some results from a tool called Metascape. It provides amazing publication-ready results for functional analysis of my differentially expressed genes. However, what they don't have is the ability to show a figure that's closer looking at a subset of the genes that are found in the enriched pathways. So let me stop for a second and make sure that generally this is making sense. So Metascape provides me this nice heat map, and it shows me in my subsets of data for upregulation and downregulation of genes in these particular condition comparisons, what pathways are involved, and right here, how enriched this in this particular condition that my genes are found. So within each one of these boxes, I know is actually a list of genes. And that happens to be my genes that are found within this pathway, which is also made up a pathway of genes. So how do I get to this step where I first have those results that you just saw? Well, first thing I got to do is create 6 subsets of data. And creating those subsets of data requires me to have fold cutoffs and other values that are going to be the parameters for creating the subset of my data. Now, I have to repeat that for each one of my pairwise comparisons. So at the end, I have 6 data sets. Now, I could use Excel to create all of those tables, and I have a big Excel workbook with 6 different tabs, but and that works for that particular analysis. However, if I'm going to repeat this again, it's nice to have a script to follow off of to go forward with my analysis. Now here we have the results of Metascape, but in a different format. So one of the things that I've talked about is accessing the data as a control point for leveraging the output and controlling it and using it for adding on to your data analysis exercise. So Metascape provides outputs in a different format than the one that we saw, which is like this. This heat map. This heat map underlying has these tables of data. One thing I'd like to point out to you is that both of these tables of data that come as an output from Metascape are tidy data sets. So that works really nicely for my tools that I use in R for data wrangling. So I now have multiple scripts for wrangling my data, and I have the opportunity now to access another set of data that I can leverage to combine to make a final figure. So I write a script, which is more wrangling, where I'm combining the output from Metascape with the results that I have in my subsets of data, combining them together and making a plot looking at my particular conditions and the relevant genes of interest that I have. And here we see that in one condition, these genes are going down, and in the other condition, the genes are going up. Now that is super powerful. And what I'm showing is that I leveraged data wrangling to do a more complex task that would have otherwise been very laborious to use some spreadsheet software to organize. So in summary, I want to review what we've talked about. We talked about wrangling data as a workflow that's an iterative process, and how that iterative process allows us to gain the deeper insight into our data and also produce a data set that is going to easily be usable for our downstream data product. You've learned about tidy data, which is a worthwhile standard to know because of all the resources available for working with it and learning how to wrangle data using that structure. And I've also pointed out that everyone can stand to gain more efficiency and value by thinking more deeply about what you're doing, even if that doesn't mean learning a scripting language. But I can help you with that, and I'd love to teach you or work with you on any of your data wrangling tasks. So I work in the Health Sciences and Human Services Library, and we have a Center for Data and Bioinformation Services. We have learning opportunities that we provide. We have resources like a high-performance computer for your data science. We have a data catalog that you can catalog your data and make records available and links for your data available. We also are interested in connecting with data users on campus and providing open office hours to work with you on any of your projects or talk about code. And we're very happy to talk to you at any time. And if you have questions, you can go to our current website that describes our resources, which is found right here as a link at the bottom of the slide. It's also worth knowing that ICTR has a voucher program that's intended to help researchers generate pilot data needed for extramural grant applications or peer-reviewed publications. And services for this program that are eligible for the support include cyber, GCRC, and also our services, where we can work on building out your project and building a dashboard, or visualizing your RNA-seq data, or running NLP algorithm and building an NLP model of your data, and gaining deeper insight and helping you get connected to your data. So with that, we're done. And I want to remind everybody to please fill out the survey that is linked in the chat box. And I'm open for any questions that you might have. Is anybody out there? Hello? Do the moderators have anything to say? Yes, I'm sorry I did not bring up SAS, but I don't discourage using any tools, as long as you're organized and in the method that you use for wrangling your data. The whole point is to be connected, to know your data, so that you can gain insight. I'm going to take a sip of water. I see, is there a training program for Linux? Well, we offer it's not going to be a Linux specific training, but we do offer insight into using command line tools and other open source tools, the type of resources that you would typically encounter when working with Linux. So happy to talk to you about that. Recommendations for starting to learn R. I highly recommend Garrett Grolman and Hadley Wickham's book, R for Data Science. That's like the quintessential data science training. And that is going to be linked in my resources guide that was on the hacking page, or the hacking slide, sorry. Yes, you definitely can find R for Data Science free online. It's a great book.
Hello, everyone. I know some folks are having questions about the online survey, and I will go ahead and add that link to the end of the chat box. So let me do that for you now, so that everyone has that right now. Does anyone else have any other questions? Thank you, Vana, for unmuting me.
I just said remember to take. I just noticed that my weird slide says remember to take the survey.
Okay, if no one has any additional questions, as JP noted, the ICTR now has included JP's services for data wrangling in our voucher program. So if you are faculty on the campus, please take a look at our website. It's the https://